this video, we will discuss the process of mammalian pronuclear fusion. It is necessary, however, to give some context to how the male and female gametes arrive in the position in the female oviduct that is best suited for fertilization, as well as how the genetic material of the gametes are able to come into contact, bind, and fuse with each other. In order to achieve this, we will consider gamete translocation, which is the transport of sperm and egg to their uterine tube, penetration of the sperm through the egg's cell membrane, the blocking of polyspermy to ensure only one sperm interacts with one egg, fertilization, such as the preparatory activation of the oocyte's metabolism, leading up to the final pronuclear fusion process.
After insemination, as the sperm enters, the uterine muscles, circled in green, helps or aid the sperm to its final destination. For this video and for your entertainment, we'll be using a Pac-Man analogy for sperm. A way to do so is to think of a fallopian tube as a Pac-Man maze. The big dot represents hurdles such as acrosomal reaction and capacitation that each sperm has to go through to be able to fuse with an egg. Within 300 millions of sperm, only the one with the highest score can fuse. In other words, the Pac-Man that are attacked by ghosts does not make it. The ghost can be used as an analogy of acidic environment, uterine mucus, immune-resident cells, and or cilia entrapment that kills millions of sperm along its journey towards the egg.
Meanwhile,
let's take a look at how this ovum was produced and ended up here.
Oogonid become diploid primary oocyte in first phase. It is completed by the third month of fetal development. Primary oocyte begin meiosis but are halted in prophase 1. Just before leaving ovary, primary oocyte completes first meiotic division and becomes a haploid secondary oocyte. This process is motivated by the follicle-stimulating hormone. Unequal cytokinesis occurs, producing a smaller polar body cell that will eventually degenerate.
The secondary oocyte is contained in a specialized Graafian follicle in an eccentric or off-center position. When released from the Graafian follicle and into the oviduct at ovulation, the ovum will contain 3 layers: oocyte, zona pellucida, and corona radiata. This is a histological cross-section of a Graafian follicle prior to ovulation. It is made up of an antrum, which is a fluid-filled cavity that characterizes a mature follicle, as well as granulosa cells that are cuboidal epithelial cells that form boundaries around developing oocyte follicles in an ovary. This is a closer look at the secondary oocyte with its surrounding corona radiata and zona pellucida. After ovulation, which is the release of a mature egg or ovum from an ovary often midway through the 28-day menstrual cycle, the ovum lives for 12 to 24 hours. Fertilization by a sperm must occur during this time for a woman to become pregnant. So as a result of oogenesis, a mature egg is haploid.
later.
Sperm capacitation is the set of physiological changes by which sperm becomes competent to fertilize the egg. This involves hyperactivation of motility and preparation for acrosome reaction. In our analogy, Pac-Man changes to a hyperactive state represented as a black quote-unquote fish. This will be important later.
The lucky few sperm who have arrived at the egg's location in the fallopian tube surround it and begin competing for penetration. When the sperm reach the oocyte, hundreds of the acrosomes erupt, releasing lytic enzymes that break down the glycoprotein matrix of the zona pellucida. The zona pellucida is composed of the 4 glycoproteins, zona proteins 1, 2, 3, and 4. Zona protein 3 was long thought to be the primary ligand for induction of the acrosome reaction, but this has recently been disproved by new imaging technologies. It is likely, however, that it is in fact zona protein 2 that is responsible for sperm zona binding. The exact molecular basis of sperm oocyte recognition in humans remained largely elusive until a major breakthrough in 2005 when researchers identified a protein receptor on the surface of capacitated sperm named Izuma-1 after a Japanese marriage shrine. The major evidence that supported this finding was that sperm that were deficient in the immunoglobulin-like Izuma-1 receptor were unable to fuse with normal eggs. Thereafter, researchers discovered the egg-binding counterpart of Izuma-1 and called it Juno, after the Roman goddess of marriage and childbirth. They were able to demonstrate that Juno-deficient eggs were able to fuse with normal capacitated sperm, proving that the interaction between the Juno and Izuma receptors is requisite for mammalian fertilization. After passage through the zona pellucida, a spermatozoan must contact, bind to, and fuse with the plasma membrane of the oocyte. Studies suggest that sperm-oocyte fusion is initiated by signal transduction pathways mediated by integrin adhesion molecules on the plasma membrane of both sperm and egg. A possible mechanism for this adhesion and fusion of sperm and oocyte may involve a receptor-ligand interaction between integrin and the vitronectin glycoprotein. Once a sperm binds the membrane receptor, its head is pulled into the cytoplasm of the oocyte.
Immediately upon the entry of the sperm, the electric potential of the egg cell membrane is changed by the production of a multiple sperm-induced calcium transient or waves.
As Dr. S suggested, the calcium wave can be thought of as a domino chain reaction.
These waves fluxes termed calcium oscillation terminates in the oocyte activation and the formation of the male and female pronuclei. The activators of calcium release stored in the sperm head is believed to be a soluble phospholipase C enzyme called PLZ-zeta. Experiments have shown that human sperm lacking PLZ-zeta cannot activate calcium oscillation in mouse eggs. Oocyte activation involves 1. Cortical granule exocytosis, 2. The resumption of meiosis I, and 3. Stimulation of maternal mRNA. A slow block to polyspermy involves the permanent mechanical removal of the remaining sperm attached to the zona pellucida accomplished by the cortical granule reaction. The protease enzyme released by the cortical granule modifies ZP2 and the other zona pellucida sperm receptor proteins such as that they can no longer bind sperm. A fast block to polyspermy has not been detected in mammals. In most organisms, polyspermy can result in a triploid nucleus or other disastrous consequences. The process of fertilization can be divided into the following 4 steps. 1. Oocyte meiotic maturation. 2. Pronuclear formation. 3. Pronuclear migration. And 4. Pronuclear fusion.
The mammalian sperm enter the oocyte whilst its nucleus is arrested in metaphase of the second meiotic division, or Met2. Calcium oscillations, stimulated by sperm entry, cause meiosis II to resume through an activation of the maturation promoter factor, which had initially arrested the oocyte in Met2, and mitogen-activated protein kinase. Inactivation of one kinase without an activation of the other induces only limited oocyte meiotic maturation. Subsequent exclusion of the second polar body from the oocyte and separate simultaneous formation of the male and female pronuclei, which occurs 4 to 6 hours after the first calcium spike, are mediated by the degradation of the substrates cyclin B1 and securin of the ligase complex called anaphase promoting complex, or APCC. As the name suggests, this complex facilitates the metaphase to anaphase transition. The female pronucleus forms when the chromosomes in the oocyte decondense and are enclosed by a nucleic membrane. Similarly, with the help of enzymes and other molecules from the ooplasm, which is the egg cytoplasm, the sperm's compact haploid chromosome set, abbreviated 1N1C, is decondensed by decomposing the protoamines that tightly wrap the DNA together. The paternal pronucleus expands and is then enclosed by a nucleic membrane.
This is an electron micrograph of the male and female pronuclei in the human ootid. Although condensed in patches, the pronuclear material appears highly hydrated. The small black sphere labeled N is the nucleolus, and some annulated lamellae are indicated by the projecting lines. In the cytoplasm adjacent to the pronuclei, numerous organelles are present, including portions of a Golgi complex labeled G.
The haploid cell that is nearly a mature ovum, produced by completion of the second meiotic division of a secondary oocyte and characterized by the presence of the male and female pronuclei, is now called an ootid.
The egg appears to destroy the paternal mitochondria by dilution and active targeting. The centriole accompanying the male pronucleus, however, is conserved and produces a star-like arrangement of dozens of microtubules from the protein stored in ooplasm called an aster. The microtubules link to two pronuclei and enables them to migrate towards each other. The process of pronuclear migration following fertilization takes about 12 hours in mammals.
Upon meeting, the nuclear envelopes of the two pronuclei disintegrate and the chromatin condenses, resulting in two sets of chromosomes. The spermatozoan centriole then divides to form the two poles of the mitotic spindle, while the egg-derived centriole is degraded. At prometaphase, the two sets of chromosomes from the egg and sperm intermix on the metaphase equator, and the common mitotic spindle initiates the first mitotic division. The first true diploid nucleus is thus first seen at the two-cell stage, not in the zygote. Our last analogy demonstrates pronuclear fusion. The Pac-Man sperm, which is now a capacitated blackfish, penetrates the egg. The two complementary haploid nuclei fuse, forming a diploid zygote represented by the harmonious symmetrical yin-yang symbol.